Okay, so let's talk about transcription. So, transcription is the process of creating messenger RNA or mRNA from DNA. So, first, give me a second here. This happens in the nucleus. So, the first step in this is the enzyme RNA polymerase. This thing right here finds a section of DNA and locks onto it with the necessary transcription factors and locks onto it. This is called initiation. Next, it unzips the DNA and copies the information to create an mRNA replica of the gene right here. Um, you'll notice that this right here is not thymine. This is uracil. That's because this thing right here isn't DNA. It's a section of RNA being created by the RNA polymerase right there. Anyway, that step is called elongation. The next step, in the next part, the RNA leaves, and the RNA polymerase leaves, and the DNA zips back together. This is called termination. Next, the mRNA then gets a 5' cap right here and a poly A tail, which is a bunch of adenines in a row. The adenines are the same thing as that right there. You'll notice I accidentally wrote T here. That is supposed to be a U, uracil. So then introns sections of I didn't add this. I didn't put the I don't have a little picture for this, but introns sections of the RNA that don't code for anything are removed, and then then you have the finished RNA and it leaves the nucleus. So yeah, that's basically transcription. Okay, so next, I'm gonna talk about translation. So translation is a process that creates proteins from mRNA. This happens in a cytoplasm. Okay, so the first thing you're going to need to know about um, all this stuff is, well, actually, there's a couple things. Ooh, that needs to be gone. Okay. One, I made a mistake. All these T's, that one, that one, that one, that one, all of them, they're supposed to be U's. I put thymine in there, and it's supposed to be uracil. There's no thymine in mRNA. The second thing you need to know, it has to do with the actual thing, is that every three letters, every three, every three is what's called a codon. Now we'll get to why these codons are important in just a minute, but just remember. And next, there you need to know about ribosome. So there's two parts to a ribosome: large subunit right here, and a small subunit. Okay, so that about does it about what you need to know. Okay, so first, mRNA finds a ribosome and is pushed in between the two subunits right here. Okay. Oh my gosh. Okay. You can see how it attaches right here. As this happens, tRNA, which is right here, well, it's that part right there, attaches to the codon. So remember codons, every 3 letters. Again, so this will attach right here. So every 3 letters codes is a codon, and what that does is it codes for a specific amino acid. So the tRNA attaches this thing. Sorry, this thing attaches to codon, which codes for that. You got it? So then this happened. So the see it attached right here. Then the mRNA right here, it goes that way, right? So everything goes through the ribosome right here. So this happens a couple more times, right? And the ribosome pushes everything through itself, right? And in the process, it rips off the amino acid, that thing, off of the top. So as this happens, those amino acids are created into a polypeptide chain. That's this thing right there. So then this happens until the ribosome reaches a stop codon on the mRNA. So there'd be a codon right over here. Right over here. I didn't put that in there. I forgot. But we'd imagine that there's 3 letters over here that tell everything to stop. So it would the mRNA would continue moving that direction until this thing went in here. Okay. So after that happens, polypeptide chain over here, you'll see the ribosome comes apart, the mRNA just goes off into the cytoplasm, and maybe to find another ribosome and be copied all over again. This happens many times. Anyway, so then these also, the ribosome also goes off into the cytoplasm, right? So now the part we're gonna want to pay attention to is this polypeptide chain here. This polypeptide chain. So it's gonna go over here. We're gonna see what it does. So what happens is the polypeptide chain is turned into, it's kind of folded into this highly complex 3D shape right here, which is a protein. So this thing right over here turns into that. So that's yeah, that's basically translation. Um, yeah.